In this chapter, we're looking at meiosis. This will be the type of cell production that gives us our sex cells, which are the egg cells and the sperm cells. And obviously, these are responsible for sexual life cycles, or they make those possible. Now, as we begin to talk about sexual reproduction, it's important to introduce the concept of genetics. Genetics is defined as the study of heredity and hereditary variation. These are things that are passed on from parents to their offspring, so these would be passed on in the form of the egg and the sperm. Now, specifically, what we're inheriting in those cases are genes. Genes are discrete pieces or units of hereditary information, which essentially boils down to this being a specific section of the DNA molecule. That DNA is going to be composed of a specific nucleotide sequence or order, and that is what our cell is going to be using to tell it how to make all of the different molecules or proteins that it needs to make on a day-to-day basis, and then also throughout its entire lifetime. If we look more at the structure of the chromosomes, each gene on the chromosome is going to occupy a specific site or a specific location, and this location is going to be called a locus. Now, if we look at a locus, the locus is shown on this diagram as this black line. So we would have specific genes located at every one of these little black marks. Now I'm going to call this particular chromosome chromosome number 1, and then I have another chromosome number 1 over here if we just apply names to these chromosomes. On chromosome number 1, this may always be gene A, located at the very bottom here. This may be gene B right next to it, and gene C here, and D and E, for example. Now, always gene A will be located on the tip of chromosome number 1. Even if we look at a different chromosome number 1, we'll always have gene A located right there at the bottom of it. Gene B is always going to be next to it. And that's what we mean by saying that it occupies a specific location on the chromosome. The chromosomes that we're seeing in this picture, notice that they look essentially the same. They're the same size and shape, and they have the same loci on them. That is because these are homologous chromosomes. What we mean by homologous chromosomes is that they're very similar, but they're not identical. So they contain information for the same biological features. This means that if gene A is a gene for eye color, both of these chromosomes have information for eye color. However, they may very well have different versions of that gene, and we'll later use the term alleles for different versions of the gene. So on this chromosome, this may represent blue, this may represent green over here. Both of them have information for eye color, but they happen to have different versions. They may have the same version of the gene, they may have different versions of the gene. Now, the reason that we have homologous chromosomes is because these come from different parents. We will inherit one of these chromosomes from our mother, and one of them will come from our father. And we get one from our mother and one from our father in the form of the egg and the sperm cells. Now, we also want to talk about what we call ploidy. Ploidy is the number of sets of chromosomes that are found in a biological cell. Or in other words, how many copies of each chromosome are contained in that cell. Now, it is possible to have one copy of a chromosome, and that's what we call the haploid condition. Haploid, it is one copy. It's represented with an N, which could be a capital N or a lowercase n. But what we're seeing here is one N. The 1 is typically not written, but it's understood to be there. If we compare this to the picture that we had on the previous slide, notice we only have one chromosome number 1. If we're talking about gametes, which are the germ cells, this is our egg and our sperm cells. These are going to have a haploid set of chromosomes, meaning they're going to have one copy of every single chromosome. So they're going to have one chromosome number 1, one chromosome number 2, one chromosome number 3, and so on. In humans, the haploid chromosome number is 23. So we have 23 different chromosomes inside a human egg cell or sperm cell. It's also possible to have two copies of every chromosome, and that's what we call a diploid cell, or 2N. This is what we had on the previous slide when we talked about the homologous pair of chromosomes. So if you have a diploid cell, you do have homologous pairs of chromosomes, or in other words, you have two chromosome number 1s, two number 2s, and so on. This is exactly what we have in our somatic cells. So all of our somatic cells, skin cells, hair cells, blood cells, those are all going to have two copies of every chromosome, and for humans, that's going to be 46 total chromosomes. Now on this slide, we're just looking at haploid and diploid cells, but it is possible to have more than two copies of chromosomes. We can have 3 copies of chromosomes, which would be called triploid. We can have 4 copies of chromosomes, and that can be called tetraploid. So we do want to keep in mind that there are other ploidies that are possible. However, when we're dealing with humans, it's really just diploid for our somatic cells and haploid for our germ cells. Plants, on the other hand, do get into a lot of those other ploidy numbers. The cell that we see on this slide is a somatic cell. Now we know that it's a somatic cell first off because it is a diploid cell. It is diploid because we have 2 copies of every chromosome. The unique chromosomes on this slide are represented as being different sizes. So we could say that this large chromosome is chromosome number 1, and we've got another copy of it over here. The medium we could call chromosome number 2s, and the small ones we could call chromosome number 3s. So notice that we have 2 copies of every single one of those particular chromosomes. That's what we mean by diploid. So since it is diploid, we write it as 2N, and then we count up the total number of chromosomes that we have, and we have a total of 6 chromosomes. And if you solve for your N, we can see that N equals 3. So those are the unique chromosomes that we have, number 1, 2, and number 3. We can talk about the chromatids in here with relationship to each other. Notice these are all duplicated chromosomes, and these duplicated chromosomes then have sister chromatids. The sister chromatids are the 2 sides of the X. They should be identical to each other in every way because one is used as the template to make the second half. We can also talk about non-sister chromatids. Non-sister chromatids are chromatids of one homologous chromosome versus its pair. Now these are not going to be identical to each other, but they're going to be very similar to each other. Keep in mind that they will contain the same loci, but very possibly different versions of the genes. Keep straight which chromatids are sister chromatids and which ones are non-sister chromatids. This picture represents what we call a karyotype. A karyotype is a picture that shows the different chromosomes that you have inside of a cell, and then those chromosomes have been organized so that the pairs are all formed, so we can see the homologous pairs. This is a human karyotype, so we do see 46 chromosomes on here. As we look through these chromosomes, they can be classified as either autosomes. So we do have in humans 44 chromosomes that are autosomes, basically number 1 through 22, and we have 2 of each one to give us a total of 44. And then we also have chromosomes which we call sex chromosomes. That's going to be the X chromosome and the Y chromosome. Now the X and the Y are not homologous to each other, and we can see first off, if you look at them, that they're not the same size and shape, and they are going to contain completely different information on them. So they don't fit what we call a homologous pair of chromosomes. If we're looking at a male karyotype, which is what we see in this picture, we have 22 pairs of homologous chromosomes. If we're looking at a female karyotype, we're going to have 23 pairs of homologous chromosomes. And that will be because females have 2 X chromosomes and no Y chromosomes. And the 2 X's that they do have are going to be homologous to each other. We want to compare and contrast asexual reproduction with sexual reproduction. Asexual reproduction really was what we were looking at in the last chapter, in chapter 12. In asexual reproduction, you have one parent or one parent cell involved, and the offspring that you get are going to be exact copies of the parent. That's what we call a clone. So a clone is genetically identical to another individual. The way that we get clones is by mitosis. Keep in mind that mitosis produced daughter cells that were identical to each other and that were also identical to the parent cell that they came from. Now, overall, that is going to be a quicker rate of reproduction than the sexual reproduction is going to be. Asexual reproduction is not something that you commonly see in animals, although there are some examples of it. Starfish can reproduce by mitosis, some other simple organisms that are invertebrates or smaller. We do see, however, a lot of asexual reproduction in the plant world. Anytime you take a cutting of a plant, that would be asexual reproduction, or cloning of the plant. Or anytime you separate bulbs, that's going to be cloning of plants. If we look at the sexual reproduction is going to require two parents. The offspring that you get are going to have a unique combination of the parent's genes. And this is also going to involve both a cell division and then a fertilization that will take place after that. So we will have this new type of cell division, which we call meiosis, that will produce the egg and the sperm cells, and then the egg and the sperm cells will come together in that process of fertilization. There's distinct advantages to both of these types of reproduction, but sexual reproduction, that second bullet there, the fact that the offspring have a unique combination of parent genes, is really why most organisms do put a bit of effort into reproducing sexually, even though that is going to be a process that requires two parents and is going to take longer. If we look at the human life cycle, you're probably familiar already with the human life cycle, but if we throw in the Ploidy terms and talk about our gametes versus our somatic cells, we all start as a fertilized egg. So if we look at that fertilized egg over here, that's what we typically call a zygote. That zygote is the very first somatic cell that composes us. It contains all of our DNA as came from our parents, from the egg and the sperm fusing together. That diploid zygote is going to divide by the process of mitosis and cytokinesis many, many, many times. And it's going to give rise to all of the other cells that make up our body. Because it is mitosis and cytokinesis that's producing those cells will be identical to each other in every way. So genetically they are the same, they have the exact same DNA content, and they're all considered somatic cells. All of them will have 46 chromosomes. Within our bodies, we have specific tissues which are called reproductive tissues. So that would be the ovaries if we're talking about a female, and the testes if we're talking about male. Because they are somatic cells, we're still going to have the total number of chromosomes of 46 in those locations. Specific cells in those reproductive organs will undergo this new process of cell division called meiosis. This meiosis is what's typically called a reductive cell division, and that's because it is going to reduce the chromosome number, and it's also going to reduce the ploidy. So we're going to start with these parent cells, which are somatic cells, which are diploid and contain 46 chromosomes, and we're going to end up with daughter cells, which are haploid, which contain 23 chromosomes. So we're cutting the chromosome number in half. Notice the top part of this diagram is haploid cells, and the bottom half is going to be our somatic cells. Meiosis is a type of cell division that, as we said, reduces the number of sets of chromosomes from 2 to 1. So it's reducing the ploidy from diploid to haploid. And it's also going to result in cells that have half the chromosome number as the original cell. So if we're talking about human cells, before meiosis, the human cells are going to have 46 chromosomes. These will be what we call our parent cells. And these are cells that will be somatic, and they're also located in either the ovaries or the testes. If we're talking about after meiosis, the resulting daughter cells are going to have 23 chromosomes each. These will be the egg and the sperm cells. We want to make sure that we understand that the egg and the sperm cells are the resulting cells of this process. They're most definitely not the cells that are undergoing the cell division. If we look at some other life cycles, meiosis and fertilization are always going to alternate during sexual life cycles. Here we have the example of the typical animal life cycle, which is the same as what we saw with the human life cycle. Most of an animal's life cycle is going to be spent on the diploid side of this equation. The only cells that will be haploid will be egg and sperm cells, which on their own cannot do anything. So notice that to go from the haploid side down to the diploid, we must have fertilization take place. So we need to get combination or combining of two different gametes. If we're going to get from the diploid side back to the haploid side, we need the meiosis process to take place, which will cut the chromosome number in half. In some organisms, we have what's called alternation of generations. Alternation of generations is when you have actual multicellular stages that are both haploid or diploid. Now, we don't have that in the case of animals, but it is quite common in plants and then also in some other organisms. If we look here, notice that there's a lot more activity going on the haploid side of this. So notice we can take a haploid cell that can divide by mitosis to produce additional haploid cells, and these are going to grow into a larger multicellular haploid structure. We can have individual gametes combined by fertilization, and then we can get back to the diploid side. Notice again that if we fuse the gametes together, we get a structure which is called a zygote. That zygote can divide by mitosis to give rise to another multicellular structure. And notice the role that the meiosis and the fertilization are playing in this. They allow the transition between the haploid and the diploid stage, or the diploid and the haploid stage. Just another variation that we can see is when we look at fungi. In this case, very little of the time is spent on the zygote side, the diploid side of this, and most of the life is spent on the haploid side. We also want to point out that both haploid and diploid cells can undergo mitosis. Mitosis produces an exact copy of whatever it starts with. So if the parent cell is haploid, the daughter cells will be haploid. If the parent cell is diploid, then the daughter cells will be diploid. However, only the diploid cells can undergo meiosis. This is because meiosis is going to cut the number of chromosome sets in half. We cannot go from one copy of a chromosome down to half a copy of a chromosome. We need to maintain whole copies. So we need to have cells that have even diploid numbers if they're going to undergo meiosis. Now, as we talk about the actual process of meiosis, this is going to be preceded by chromosome replication. That would be in the form of an interphase, which will be identical to the interphase that we had before. And then we're also going to have two actual cell divisions. We're going to call those meiosis 1 and meiosis 2, and they will be dividing different things. The end result of this process is that we should get 4 daughter cells, and each cell is going to have half the number of chromosomes as the parent cell. So we're going to take a cell, one parent cell, we're going to split it once in meiosis 1, and then we're going to take each of those and split them again. So this is what we mean by our 2 divisions. This is where the 4 daughter cells will come from, and each of those daughter cells is going to be egg cells or sperm cells, depending on whether we're talking about this process taking place in the ovaries or the testes. If we look at an overview of this process, In interphase, we will start out in G1 interphase, similar G1 to what we had before, and notice that these are chromosomes composed of one chromatid each. Now, we're going to take those, and through the S phase of interphase, we will copy all of them such that we end up with duplicated chromosomes in G2. Those duplicated chromosomes will be composed of sister chromatids that are identical to each other in every way. And then we also have the non-identical sister chromatids there as well, which would be the non-sisters. If everything checks out okay, we'll get the signal to go on and divide by meiosis 1. In meiosis 1, the homologous chromosomes are going to be pulled apart and separated from each other. When this happens, we do result in haploid cells. They are haploid cells because they only have one of that particular chromosome inside, even though it happens to be a replicated chromosome. In meiosis 2, we will take the sister chromatids and those will separate from each other. When we finish meiosis 2, we will also have haploid cells, but now these are going to be haploid cells composed of one chromatid each. So if we run through here, in G1, these will be non-replicated diploid cells. In G2, they will be replicated diploid cells. When we're looking at meiosis 1, when we finish meiosis 1, these are going to be replicated haploid. And then if we're looking at the very end product, these are going to be non-replicated, and they'll also be haploid cells. Now, because we have two cell divisions, we do have twice as many phases as what we looked at in chapter 12, but we're going to go through these one by one, talk about what's happening during the phases, and how to recognize the cells. If we look at interphase, a cell in interphase is not going to look any different from a cell that was in interphase in the last chapter. That's because we are doing the same thing. We've got everything being copied inside the cell, and we're using the DNA. So the DNA will be unwound, which means that we won't really be able to see any chromosomes. Now, at this point in interphase, the chromosomes have already been duplicated, but as we said, they're uncondensed, and they will be composed of two genetically identical sister chromatids. As far as the ploidy and the replication status of this cell, at the beginning of interphase, so this would be talking about G1, these are going to be diploid cells, but they'll also be non-duplicated, meaning that they're composed of chromosomes with one chromatid each. By the time we finish interphase, these are going to still be diploid cells, but they're going to be diploid duplicated cells. So at this point, our chromosomes will be composed of two chromatids each. If everything checks out okay, then we will get the signal to continue on into meiosis 1. In meiosis 1, the homologous chromosomes are going to get separated. So that is the overall goal of meiosis 1. We will start off with a prophase, which we call prophase 1 because it's part of meiosis 1. And this really is the most important of all of the meiosis phases. Over 90% of the time of meiosis is going to be spent in prophase 1. So there's certainly a lot of things going on. We will have the same things going on that happened in the mitosis prophase, such as the condensing or compacting of the chromosomes. This means that they will become visible. We're also going to get building of the spindle apparatus. So our centrosomes will be moving apart from each other, moving to opposite ends of the cell, and the spindles will be growing out of them. Now, if we talk about unique things that are going to take place, the homologous chromosomes are going to find each other. So this process will be called synapses. The two number 1 chromosomes will find each other. The two number 2s will find each other. The two number 3s, and so on. If we're talking about a female human cell, we're going to have 23 pairs of chromosomes that need to find each other. When they find each other, they are going to synapse or pair up with each other, and they will line up and physically connect with each other such that all of the homologous or all of the loci end up across from each other. Once they do that, we have this structure formed which is called the tetrad. The tetra part, which tetra means 4, that stands for the fact that we've got 4 chromatids lined up with each other. It is still considered 2 chromosomes, but it's 4 chromatids in 1 tetrad. Once they've accomplished the tetrad structure, then we're going to have crossing over take place. Crossing over is an exchange of non-sister chromatids. This means that we're going to actually break the chromatids and they will swap pieces with each other. So notice here that we're going to end up with chromosomes that are now part red and part blue. The actual structures where the non-sister chromatids overlap with each other are called chiasmata. And the significance of the careful synapsis was that in this case, if we break in the same place and swap pieces, we are swapping the same loci for each other. This means that no chromosome is losing information and none is gaining information. We're simply swapping versions of the information. Now, this crossing over process takes quite a bit of time, which is really why we have such a high percentage of our time spent in prophase 1. Now, as far as the ploidy and replication status of this cell is still going to be similar to the G2 cell. So in this case, we've got a diploid cell and the chromosomes are duplicated. If we're trying to recognize a cell in prophase 1, we will be looking for chromosomes that we can see, and then we also want to see those tetrad structures formed. Not only do we want to see the tetrads, but we want to see them scattered about inside the cell. Once that is accomplished, we're going to move into a metaphase. And this is metaphase 1, in which case we will be lining things up on the equator of the cell. But when it happens, these tetrads will still be held together. So metaphase 1 looks quite unique. We should be able to see the tetrads lined up on the metaphase plate. The ploidy and the replication status will be similar to what we had before. It's going to be duplicated and it is going to be diploid. In anaphase 1, we are separating the chromosomes. We're separating the homologous chromosomes from each other. Another way to state it would be to say that we are pulling apart the tetrads. In this case, now we've got homologous chromosomes moving towards the 2 sides of the cell, and the sister chromatids are still going to be stuck together. However, notice that the sister chromatids are no longer identical to each other at this point. If you look at the ploidy and the replication status, this is going to be a diploid cell that happens to be duplicated still. Now, anaphase 1 should look quite unique as well because we have replicated chromosomes, or X's, that are actually moving towards the 2 poles of the cell. This is different than what we had moving to the 2 sides when we talked about mitosis anaphase. In telophase 1, we're going to be rebuilding. Now, we're going to very quickly go into a meiosis 2. Since we will be continuing on into a meiosis 2, we will not completely rebuild everything because we would have to turn around and take it all apart. So we may or may not get the chromosomes unwound. We may or may not get the nuclear envelope rebuilt. But we will take apart the spindle apparatus because we will have to rebuild that. Notice that at this point, each chromosome is still composed of 2 sister chromatids. Cytokinesis is going to take place as well, and if we assume that cytokinesis has occurred, the ploidy and replication status of each of these cells is going to be haploid, and it is going to be duplicated. So notice that each of these cells has only 1 large chromosome and 1 small chromosome, and the chromosomes are always composed of 2 chromatids each. After telophase 1, we're going to proceed right into meiosis 2. Notice there is not going to be a separate interphase in between meiosis 1 and 2. This means that we will not copy the DNA a second time. In meiosis 2, we will be separating the sister chromatids. At this point in this process, these are going to be non-identical sister chromatids. And you will find that meiosis 2 is very similar to mitosis. In mitosis, we were separating sister chromatids as well, but they happen to be identical sister chromatids. In prophase 2, we are doing all of the same things that we did in prophase of mitosis. We are forming the spindle apparatus, condensing the chromosomes if need be, and taking apart the nuclear envelope. We would also be attaching the spindles to the kinetocores of those individual chromatids. Now, as far as the ploidy and the replication status of this cell, this is still going to be a haploid cell that happens to be duplicated. In metaphase 2, we are lining up the chromosomes along the metaphase plate. So now we've got X's on the plate, similar to what we had in mitosis metaphase. The ploidy and the replication status has not changed, so we're still looking at haploid and duplicated. In anaphase 2, we separate those non-identical sister chromatids from each other, so we see them moving towards the two poles of the cell. And the ploidy in this case is going to change a little. So this is the tricky one. At this point, it is going to change to a diploid cell for just this one stage. Notice that we do have 2 of the large chromosome and we have 2 of the small chromosome. So it will be diploid, but it's also going to be non-duplicated, because notice that these are no longer X-looking chromosomes, or chromosomes composed of 2 chromatids each. In telophase 2, now we really are going to have a full-blown telophase. We are done with the meiosis process, so we will completely reform the nuclear envelope. We will decondense or unwind the chromosomes, and the cytokinesis process is going to take place to divide these into 2 new cells. If we look at one of these cells, then we can see that these are going to be haploid cells, and they're also going to be non-duplicated cells. Keep in mind that these cells that we have, these are our daughter cells. We would have a total of 4 of them, and they would be either egg cells or they would be sperm cells. Notice in this meiosis process that we went through, the original cell started with 4 chromosomes, and we're ending up with daughter cells that have 2 chromosomes each. So we have cut the number of chromosomes down. And also notice that we started with a parent cell that was diploid, and we're ending with daughter cells that are haploid. That is the goal of meiosis, to reduce the number of chromosome sets down and cut the chromosome number in half. In humans, we will start with 46 chromosomes and we'll end with egg and sperm cells that have 23 chromosomes.